Great. Good morning, everyone. Following disappointing phase 3 data on the cardiac cell therapy product C-Cure last summer, Celyad is now focused on oncology and developing novel CAR products. NKR-2 is in a phase 1 study in multiple indications, and here to present for Celyad is George Rawadi, who's the Vice President of Business Development and IP. George, thanks for joining us today.
My pleasure.
And I think the way we're going to do this one is a little bit more of a dialogue, and I'll ask you some questions, and we'll have some supporting slides to answer. So, George, maybe you could start off by discussing Celyad's lead CAR-T product, which is called NKR-2. And you just published a very informative white paper, actually, that can be found on the company's website, a little pitch there. It's really an excellent piece on that. So how is NKR-2 different than other CAR-T constructs that are in development and other ones that we'll hear about today at the conference?
Yeah, it's, well, first, as everybody knows, Celyad, it's a 10 years old company. We've been initially developing cell therapy for cardiac disease that we took from preclinical stage all the way up to phase 3. So throughout this development of the cardiac disease, actually the company developed significant skills in cell therapy in general and in manufacturing in particular. And now we are leveraging all this expertise into our CAR-T program. So what is quite specific and unique about the CAR-T platform that we are developing, that it's actually, instead of using, I would say, the classical approach of CAR-T, starting from a tumor antigen, antibody targeting this tumor antigen in a specific disease, and then constructing a chimeric receptor using the binding moiety of this antibody. Instead of this, we are using actually NK cell receptor that are naturally occurring and expressed on NK cells, and they have been, well, optimized by nature to bind to a set of ligands that are expressed on tumor cells. So we use this basic biology of the NK cells, and actually we make those receptor chimerics with some minor changes and express them in T cells. And this is the particularity of our platform. Here on the slide, we show the construct of our NKR-2, our lead program, which use NKG2D, fully human NKG2D, that has been made chimeric by the adding of CD3 zeta signaling domain. So this add a first signaling cue to the chimeric receptor. And the other particularity is that NKG2D naturally also dimerized with a signaling molecule called DAP10 that is also expressed in T cell. And upon the expression of the chimeric NKG2D, it associates with DAP10 and provide with a second signaling. So both CD3 zeta and DAP10 provide two signaling mechanisms upon binding and activation of chimeric NKG2D, which is very close and look to the second generation of CAR-T.
So really, it's interesting because the natural killer receptors offer broader targeting in a range of cancers, and then by fusing them to the T cells, that's how you're infecting the kill, essentially. So at ASH last December, you reported data from your first clinical experience with NKR-2, and this was a phase 1 study in both AML and multiple myeloma patients. What did, what doses did you explore in that phase 1 study, and what did you learn from this patient experience?
So indeed, our NKR-2 CAR-T has been gone through a first phase 1, and the main objective of this phase 1 was safety and feasibility. This was the first time NKG2D is ever used in a clinic, so we were very cautious in the dosing that we were using. So we started really at a very low dose. This was a dose escalation study that was completed at the Dana-Farber Cancer Institute here in Boston that explored 4 cohorts at increasing doses from 1 million up to 30 million of NKR-2 CAR-T. This looks like really low, but this was done intentionally to just be on the safe side. So what we have done through the study is to establish a great safety profile of NKR-2, and in the same time we've been, I would say, nicely surprised to see some unexpected clinical activity, where at the highest dose we were able to observe a prolonged survival of those AML and multiple myeloma patients. Those are relapsed and refractory patients.
That's great. And now, based on those results, you've initiated the phase 1b THINK study of NKR-2, both in Europe and also the IND was recently cleared here in the United States. So what doses will you be using in this study? And I know you're expanding to much broader tumor types, so maybe you can walk us through the different types of cancer you'll be evaluating as well.
Indeed. So now THINK, it's a follow-up study to the first safety study. That is currently enrolling in both Europe and US. So we have 3 sites in Belgium that are enrolling versus study. We have 2 sites activated and enrolling in the United States, and there are a third additional site that will be activated very soon. So the THINK trial is actually building on the first safety trial that we have completed. The doses are much higher than in the first safety study. So here we are starting from 300 million going up to 3 billion of cells in a dose escalation THINK study. And instead of doing one single administration of our NKR-2, we are actually performing 3 subsequent administration every 2 weeks in those patient population. So we are, in terms of diseases, we are first continue targeting AML and multiple myeloma. And because of NKR-2 target set of ligands that are expressed on multiple tumors, we are also taking advantage of targeting solid tumors. And based on immunohistochemistry data, we've selected 5 tumors where the receptor are highly, the ligands, sorry, are highly expressed, and this is ovarian, colorectal, bladder, pancreatic, and triple-negative breast cancer. So, as I mentioned, THINK now is enrolling. We will have a first phase or segment that is dose escalation, and when we would have identified the recommended dose, then we will specifically expand in each of those 7 indications.
And with such a dramatic increase in the number of cells and going from single administration to multiple administration, are there any extra safety parameters that you're keeping a close eye on those first patients during those first few weeks?
Well, the THINK trial actually, or the protocol, is mimicking where we see the highest efficacy in preclinical models. So in the preclinical models, the highest efficacy we see is at multiple administration at doses that are in the range of the ones that we are currently using in THINK trial. And let me just explain why we are looking for a repeated administration. Actually, NKR-2 is designed not to persist in the patient and not to massively expand. So that's why we see it more as a drug and then as a, I would say, engrafting cell therapy. So that's why we are going for three administration, which have also a safety aspect built into that, because the cells will not persist for a long time, and we don't have to worry about a long-term safety concern.
And George, when could we get preliminary data from this THINK study?
So now we have started the THINK trial. It's an open label study, so we'll be able to report on the progress of the trial as we move forward. The dose escalation is expected to be completed by end of the summer, toward August, September. And then at that time, we will start the expansion phase. The expansion can go much faster than the dose escalation because we have much less, I would say, freedom in terms of pace of enrollment and infusion. And we expect to have the top-line data from the full study by Q2 2018, which is about a year from now.
Excellent, great. And before we transition to some of the other contracts, are there any questions on NKR-T? So, George, with the time that we have remaining, what are the next NKR constructs that you are developing, and how are these different than NKR-T?
So first, we are developing NKR-2 allogeneic. We have an allogeneic technology that is different from the gene editing and that is very appropriate for the type of NKR-2 cells. So this is today still in the preclinical stage, and we are looking to start our clinical program with the NKR-2 allogeneic by mid of next year. On this program, we have also a license agreement with Ono Pharma in Japan, Korea, and Taiwan, where they obtained exclusive right to the NKR-2 allogeneic. On the top, we are working on what we call NKR-2 beta, and this is keep leveraging on the same construct, but giving different features and different characteristics to the T cell to do just more than the cytotoxic and the killing activity. It's too early now to comment in detail on that.
That's helpful. So you mentioned the allogeneic program. The USPTO recently rejected a re-examination request of your allogeneic CAR-T patent. Can you explain what this is and how important this is to Celyad?
So at the time of the acquisition back into January 2015 of all the CAR-T portfolio and IP that we are developing now from Dartmouth College in New Hampshire, there is a set of patents around the concept of allogeneic CAR-T that were filed, I would say, early on before the big buzz around the CAR-T and allogeneic CAR-T. And these patents claim the concept of making allogeneic CAR-T cell without being specific on some methods to generate them, but the how to produce, how to manufacture, and how to treat cancer patients. And this was granted by the USPTO. It has a significant implication in terms of exploitation of this kind of technology by third party, and the patent has been challenged several times at the USPTO, and the USPTO held it without any changes, which shows the solidity of our patents. So the patent gives us the freedom to operate on our allogeneic platform, and definitely give us an opportunity also to provide licenses to third parties that are also developing allogeneic CAR-T. And we are in discussion with some of them today.
Excellent. George, that's very helpful. I'm going to see if there's any questions from the audience. I want to thank you very much for being here with us today. Oh, was there a question? Sorry, please go ahead.
Natasha Bogosian. So given that you have a quite robust autologous platform, and although your allogeneic is in preclinical, what is the major driver for adding the allo? Is it really cost of goods, manufacturability, or are you seeing a difference in efficacy? And is there any concern on Celyad's side that's going to end up being cannibalistic to the allo? Or are you going after different indications?
I think it's a good question. For us, the important is to build optionality in our clinical development. Today, definitely autologous is the safest way and the fastest way from regulatory perspective to go and approve safety and the efficacy of any CAR-T. Definitely, it has challenges in terms of manufacturing when it goes to a large indication, which are not niche product or specific diseases. So hopefully, allogeneic will be able to address that with still its own challenges from regulatory perspective, from banking, sourcing of the cells, etc. So the idea for us is to give us optionality, and we will develop what we believe has a better chance from business perspective to make a success.
Excellent. George, thank you very much for being with us today. We look forward to more data readouts.